not infiltrating myeloid cells, is upregulated. You can see a series of tumors here are much lower. And this paper actually just in press as of a couple days ago, so this will be coming out. But the PD-1, PD-L1 pathway is important, and it was asked yesterday, where is the PD-L1? So last year we published a series of tumors. This is double staining. You don't really need to know which one was red and which is brown. This is PD-1 and PD-L1. What I want you to appreciate is that there are at least 5 different patterns that we could see where they are either totally disconnected, it seems like they're independent events, or down here, I'll blow it up because you can't see it, where it seems that there's an actual synapse, where you see PD-1 and PD-L1 almost lining up like an army against the battle gates. In the PD-1, PD-L1 synapse in this sort of a tumor, perhaps 1 out of 20, 1 out of 5 is 20%. Perhaps these are the sort of tumors that we need to be looking for patterns, and these may mediate the response, because this looks like PD-L1 is an effector turning off the PD-1 positive T cells. So I think the distribution in counter staining for PD-1 and PD-L1 could be valuable now that these assays are worked out. One thing that jumped out at us, it's very obvious these days when somebody from the lab comes down with some tumors, I say, okay, does it look different in HPV positives and negatives? And this sort of jumped out at us that there are higher frequency of PD-1 positive T cells in the TIL of HPV positive patients. So that didn't make so much sense. We've got a good prognosis group, but they have more of these exhausted cells than the HPV negatives, who still have a higher amount in the TIL versus the peripheral blood, but lower than the HPV positives. And I'll try to give you a sense of how we've worked that out to explain this somewhat dichotomy. This is one example. So I made this point yesterday. John Wary in the mouse study had shown us that the extent of PD-1 is really the critical determining factor. So we shouldn't think about PD-1 as a binary positive negative. What we did was we took tumor infiltrating lymphocytes that were CD8 positive, and we segmented them by PD-1 high, intermediate, and low. The high is only about 2%. But if you now looked at the prognostic impact in HPV positives versus negatives, the HPV positives had very few of the PD-1 high. Most of the PD-1 positive cells were low to intermediate in the HPV positives. And the reason that's important is that you can see down here, the PD-1 high make almost no interferon gamma. They're very difficult to activate. Interestingly, they have plenty of granzyme B. This is consistent with other exhaustion data that we have in TIL, that granzyme is retained, but the ability to make interferon gamma and function as a type 1 CD8 T cell is dramatically impaired, and this is due to the PD-1 high. So that's, I think, the marker of exhaustion. The HPV negatives had lots of the PD-1 high. Perhaps that's the poor prognostic impact in these patients. Our statistician was able to give us a little bit better way to portray this, and you can see the relative risk of recurrence is dependent on the extent of PD-1 highs, and you can just follow the hazard ratio increase as the fraction of PD-1 high goes up, and the risk of recurrence goes down if you have lots of PD-1 low to intermediate. These are the activated cells, and so an anti-PD-1 strategy should really clarify for us whether you're enhancing the PD-1 low to intermediates, which are associated with a good response. What is PD-1 doing in head and neck cancers? We isolated tumor-infiltrating lymphocytes from a series of tumors, compared the frequency of this type 1 pathway that we know is strongly anti-tumor, characterized by phosphorylation of STAT1, the transcription factor TBET, that is a strong Th1 and a good anti-tumor, and then phospho-S6 downstream of the mTOR pathway, and also not only the T cell receptor, but also the TIM3 pathway, a second checkpoint receptor that I'll show you some data for later. But you can see these are all suppressed in the microenvironment associated with PD-1. PD-1 suppresses STAT1, TBET, and phospho-S6 through the sarcomology-containing protein 2. SHP2 can dephosphorylate STAT1. And so you have a pathway where PD-1 overcomes activation from the T cell receptor, where you need to have good activating pathways. And this dephosphorylation event through SHP2 is highly upregulated in PD-1 positive TIL. And so we need to reverse this. We can do this in the lab. You can see that all of these features can be driven lower This third set of bars is with a PD-L1 coated bead mimicking a tumor cell. So you coat a bead with PD-L1 like the tumor, and you can drive down phospho-S6, TBET, and STAT1, and this is able to be recovered when you use an anti-PD-1 antibody. So you can drive the microenvironment away from the Th1 type 1 anti-tumor activity and reverse that with an anti-PD-1 antibody. So does that matter? Here's a Tanguy presentation. You can see a frequency of response rate of 20 to 25%, two different cohorts here. And as he alluded to yesterday, in one of these, the HPV positive did slightly better. In the other, the HPV negative did slightly better. But roughly, we're looking at a 20 to 25% response rate. This is from a discussion that our medical oncologist, Julie Bauman, presented, where she compared this to the current FDA-approved agent, single agent cetuximab, which is about half of the response rate in a heavily pretreated second-line cohort. And so this was quite appealing. Anti-PD-1 works. This has led to a randomized phase 3 trial, which completed accrual last summer. And this was in a platinum refractory, second-line cohort. So we're anxiously awaiting those data. And this was compared to standard of care, which is not always cetuximab. Some of these patients, I think, got methotrexate and a taxane. So these data are incubating. We know that durvalumab has activity. This lady had no idea she was going to be so famous, but she gets shown very frequently, and it's nice to see the smile on her face as this big tumor, clearly an oral cavity, HPV negative tumor, was regressing. But the key thing here is interesting is that older folks get the same relative risk reduction benefit than younger folks, and this is maybe where the future is going to go. What do we want to use? Systemic toxic chemotherapy? Well, we know now we can use immunotherapy, perhaps because our lymphocytes become senescent through the PD-1 pathway as we age. You can see some of these responses. This is PD-L1 negatives, PD-L1 positives, and the waterfall plot shows a little bit enrichment in the PD-L1 positives as shown here in the spider plot. And we do need to pick up these partial responders and look for escape mechanisms for de novo resistance versus acquired resistance and get post-treatment specimens. So PD-1 is just a single, a second signal, but there are others. There are these positive signals as shown in green. There are inhibitory negative signals. I made the case yesterday that PD-1 is generally felt of as a cell intrinsic suppressive mechanism because PD-1 is present on the T cell itself, but there are cell extrinsic mechanisms mediated through CTLA-4 and regulatory T cells in the microenvironment. We know about myeloid-derived suppressor cells, and so targeting cell intrinsic and extrinsic, I think conceptually helps us understand why these two agents should be additive if patients are dependent on each mechanism. So AZ has put together a series of trials looking at a combination of PD-L1 targeting with CTLA-4 targeting, both in the PD-L1 positive as well as the PD-L1 negative subgroups, and then the confirmatory phase 3 trial will segregate and stratify PD-L1 positives versus negatives and look at the combination of PD-L1 with CTLA-4 targeting versus PD-L1 targeting alone or standard of care. So a series of trials here with combinations as we get to the cell intrinsic and extrinsic strategies. The theory, the hypothesis for this was driven by lung data, which was probably presented a couple times here, that you can see that the PD-L1 negatives have a relatively low single agent anti-PD-L1 activity, but when you combine it with CTLA-4 targeting with tremelimumab, you get a very nice response. So in head and neck, that's really what we're hoping for in those PD-L1 negatives. So we wanted to ask the question, as I mentioned to you, what is the best approach for therapy? So we developed a mouse model here where we use anti-PD-1 therapy with radiation therapy. And I made this case that PD-1 high are bad. Those are the exhausted cells. So we wanted to then take that into a in vivo model prior to testing in precious clinical trial specimens and ask, whether anti-PD-1 therapy modifies these PD-1 high versus the activated intermediate cells. And you can see that here's anti-PD-1 alone, here's radiation alone, and the combination. And so combining this in locally advanced head and neck cancer makes sense. And if you look at the best clinical response of anti-PD-1 plus radiation therapy, the PD-1 high cells disappear, and you have a nice dramatic increase of the PD-1 low to intermediate, the activated cells. So it looks like we need to make more of these activated cells and get rid of the exhausted PD-1 high cells. And this may help us to figure out who's likely to benefit based on what the extent of their PD-1 high exhausted cells are in the clinic. I've talked about T cells. Let's not forget that natural killer cells, which are important for the use of therapeutic monoclonal antibodies like cetuximab, can also be activated and express PD-1. And so the effect of anti-PD-1 may not simply be on T cells, and you can see that NK cells express lots of PD-1. There's an increase of NK activation and PD-1 expression. Now, the difference, as I just told you, PD-1 high on T cells is bad. When we looked at PD-1 high on natural killer cells, that was a good prognostic factor. So there's some biology we need to understand of different T cells, NK cells, different effector cells in the tumor microenvironment, because the NK cells are likely to interact with therapeutic IgG1 tumor-targeted antibodies like cetuximab and increase their activation and PD-1 positivity. And so we shouldn't just transfer the information we get from one cell type to another, since they behave differently. So the locally advanced setting is going to add anti-PD-1 like our mouse model to chemoradiation and compare that hopefully with cetuximab radiation, the other FDA-approved regimen in locally advanced head and neck cancer. This will be discussed further by Dr. Vogt, so I don't want to go into too much detail. But I told you that there are multiple checkpoint receptors, so I want to talk a little bit about PD-1 inhibition in the setting of another molecule called TIM-3. This is a promising target for data that I'll show you. When we looked at the tumor-infiltrating lymphocytes, the PD-1 positives on the y-axis, about half of them express this second receptor called TIM-3. And what we found simultaneously with the other PD-1 high and intermediate was that the TIM-3 positives have the highest PD-1 expression, and the TIM-3 negatives are those low to intermediate. So we wanted to ask whether these cells are more or less exhausted. So I've taken that graph. You can see here, based on the bars, the interferon gamma secretion here at the protein level and the RNA level, now looking at exhaustion markers, that these double positive cells have the highest level of exhaustion markers of LIMP1 and BATF. And you can see the PD-1 intermediates make lots of gamma, and these are TIM-3 negative, suggesting that targeting TIM-3 plus PD-1 would be therapeutically beneficial. Interestingly, TIM-3 single positives proliferate very nicely. So there's some biology we need to understand, because TIM-3 does not seem to be a single exhaustion marker, since if you have PD-1 negative but TIM-3 positive, they actually proliferate quite well. But when TIM-3 is present with PD-1, the proliferation is dramatically impaired. So we're actively studying these two subgroups and have a recently funded grant to look at combined targeting. What was in the literature was that targeting PD-1 plus an anti-TIM-3 antibody led to better anti-tumor activity. So we asked, well, what if you use just single-agent PD-1? What happens to TIM-3? And so we went back to our mouse model that I showed you and looked. Actually, this is just in vitro, preliminary to the mouse model. And what you can see that if you use an anti-PD-1 antibody, the levels of TIM-3 actually go up. This is just in vitro, a short 24-hour window of anti-PD-1, and you get enhancement of TIM-3 expression. So there may be some compensatory upregulation of a second checkpoint receptor. So then we went to the mouse model. We looked after anti-PD-1 antibody, and there's a really much more dramatic upregulation of TIM-3 when you use a single-agent anti-PD-1 in this model. And you can see expression both in CD8s and CD4s, where TIM-3 expression goes up. And this correlates with the partial responder status of TIM-3 upregulation now in the splenocytes from these anti-PD-1 treated mice. So in the circulation and in the tumor, in a partial response with a single agent anti-PD-1, a second receptor is trying to potentially escape and upregulate its expression to mediate exhaustion. So targeting the tumor microenvironment, reversal of immune escape is what we're trying to identify. We have to turn the clock back and figure out whether the key is that the tumor cell cannot present antigen, whether there's no signal 1, to figure out whether there is aberrant co-inhibitory signal 2, whether there is a poor signal 3 of cytokine secretion and inflammation, and then others have talked about signal 4 being the chemoattraction into the tumor microenvironment. But the key is to reprogram these anti-tumor T cells and make the tumor a better antigen presenting cell. There's the lab. Thank you.